Hello, everybody. Can everybody hear me?
Yep.
Yep.
Yes.
Right.
Yes, thanks.
I do have to say, a quite peculiar legality in that if anybody, feels they are being identified in the video, I can't really post it, publicly. So you either have to change your name or log out, but, none of you is actually showing the images at any rate. So I'm just going to make you aware that, for privacy reasons, I have to give you that little disclaimer. I also need to say that it's kind of an interesting experience. The reason I put the video on YouTube is quite simple. If I were to put it onto the university system, they have some sort of software which compresses the file very, very strongly. And I think lots of our images are very visual, very attractive, and I don't really want to actually lose that kind of impact on the visuals. Speaking of which, in the last lecture, we finished off that we said we can't really understand rocky shore communities without actually understanding larval biology. The reason for that is all those sessile animals have larval stages which go out into the ocean, disperse, and come back onto the shore. So that basically was a very, very nice segue to today's lecture, which is all about the plankton. And just before the Easter holidays, I thought we make it a little bit more relaxing. So it has more short YouTube videos in it than the other ones. And the reason for that is actually quite peculiar. That if you actually write a lecture, like I do every year, and I try to keep it actually a little bit different every year, and you look for material for rocky shores, for sandy beaches, even for the oceans, you don't actually get really any captivating imagery. If you were to go and look for plankton, and you will see that today, there's actually a lot, because there's an army of people out there, and they basically, go into, any part of the sea or estuary or lakes with a net, scoop it out, buy themselves a microscope, and they have it as a hobby, because those organisms are visually very appealing. And so today, we basically borrow a lot of that material, and I kind of interject in between. Now, starting with that kind of premise, our first, video, if we can actually get into it, is basically, exactly that, and it gives you a little bit of a journey of a planktonic organism. So can we get that going? Yep, here we go. I basically need to disappear. Can I disappear? Yeah, here we go.
We all know there's more water on Earth than land. Just in case you don't, the seas cover 71% or... So you can't...
So you got kind of the drift from a perspective of the ecological importance of it all. Hang on, hang on. Sorry for that. I'm trying to get right. So before we actually go and talk about the plankton proper, I have to actually step you through some terminology. And I know there's a lot to be taken in now, but view those slides, those next 10 slides, as something you should actually have throughout your studies. And I particularly spent some time putting that together, because when I was a student, none of our lecturers, our professors, told us the terminology. And essentially, it was a language, which basically evaded me for many, many years, and I think many other students suffered as well, simply because you get so used to it that you don't actually bother explaining it to other people. And that kind of language reminds me when I try and fix, the Land Rover, when you get, a Land Rover manual, it's all about, basically you say, oh, take that pin or take that pin. And it doesn't actually make any sense really, because the people didn't actually bother to translate the technical jargon to everybody. So the first thing we have to be quite aware when we talk about plankton, yes, there's plankton in freshwater systems, but overwhelmingly we're talking about marine systems. Why? Well, when I was your age, somebody said to me, like, one professor said to me, yep, young fellow, don't worry about, studying freshwater systems. They're but a rounding error when we sum up all the water on Earth. And that's actually probably quite true.
Was gonna like
Hello?
in the middle thing. Oh, it's like this. Hi, baby.
Hello, Isabella. So essentially, when we talk about, aquatic biology at a global scale, it's all about the oceans. Freshwater is about 2.5%, but it all sits basically bound up in the ice masses. So particularly in a country like Australia, I mean, really, don't really bother about it. And here is another slide to actually show you pretty much the same thing. Now, what is kind of, astonishing though is, can I see myself? No. More, is that essentially the two fields of aquatic biology, the one being marine biology or oceanography, and the other one freshwater biology or limnology, are quite simple. You can actually make a little decision tree. So if the water contains salt, we're talking about marine stuff. If it doesn't, it's freshwater. What is actually sometimes not as clear is that when people say, ooh, we're actually talking about coastal or neritic processes as opposed to offshore and oceanic. And you might actually ask yourself, well, where does the oceanic province start and the neritic, province stop? The answer is actually quite simple. It's actually at the shelf edge. And we can actually determine that shelf edge quite simply nowadays. You can just go and look at Google Earth. Hang on. So essentially, here we have, if you go out from Mooloolaba, whoops, let's go on back, about 30 miles, there's a very, very distinct shelf edge where it goes from about 40, 50 to 100 meters right down to about 4,000 meters. You might say, well, that's only basically in Australia because we're an island. We see all the awful news of New York. We were supposed to go there to work on New Jersey beaches about here for about a month in June. Of course, that's all cancelled now. But it's also very, very distinct here. You get a neritic province and you get an oceanic one, and it all basically divides here at the shelf break. Look at this cool canyon going down here. Isn't that cool? Anyways. And we get the same, up further north, if you go off Fraser Islands. Very, very, sharp distinction. And of course, there's the city of Hervey Bay. It's not quite New York, but you get the same thing. And that's sort of a false color image. You get out to about 50 meters, and literally within a half a sea mile, nautical mile, you go all the way down to about 3,000 or 4,000 meters. So this is basically the terms when you hear about neritic and oceanic. They are, distinguished by the position to the shelf break. Now, essentially, if you actually classify marine life, you only have 3 different types of habitats. You get stuff which actually swims or drifts in the water. So it's basically habitats in the open water bodies. Then, technically speaking, we can take the interface between the bottom sediments and the larger water column, which is basically, maybe half a meter or so. And then it's the space between the particles within the bottom substrates, basically that's everything which lives on the seafloor. If you actually, look at a marine biology textbook, I think they're always incredibly funny. I'm actually at the moment writing one. That's the good thing about lockdown. So everybody has to buy it next year. And I'm not quite sure whether I'm going to use those kind of images. There always has to be a turtle. I don't know why. And a shark and a dolphin. But look at the size of this kind of squid. It's enormous. It's the size of a shark. Never mind. You basically differentiate between stuff which, swims or drifts in the water column called the pelagic zone, and the benthic zone at the bottom. However, it goes a little bit further, that essentially in the water column, the noun, the collective noun is called the pelagos. All of those terms come from Old Greek, whereas the collective term for the organisms, is the pelagic. So you would actually talk about a pelagic shark or a pelagic fish or a pelagic jellyfish. Those are organisms which do not normally, live anywhere near the sea floor. If you go down to the sea floor, or if you really have to, doing it in freshwater lake floor, to the bottom of the ocean, or any freshwater systems, you don't look about the pelagos, it becomes the benthos. So essentially the benthos is the collective term of every single living bit sitting down at the bottom, and we talk about benthic organisms which are associated with the seafloor as opposed to pelagic ones. You can then actually go a little bit nitpicky and say, well, if it's at the surface of the seafloor, it's the epibenthos. If it's buried inside the sediment, it's the endobenthos, and so forth. Now, this looks actually very, very horrible, but it's actually something you might actually want to keep. Took me quite a while to actually put that together. But essentially, if you then, we're talking today about the pelagos. Now, the pelagos are pelagic organisms, which means they are living 99.9% in the water column. They go down to the bottom occasionally, but usually not. Then you can actually differentiate how they get the energy. Either you are a plant and you make it yourself. The complicated term is called a photosynthetic organism or primary producers. Whereas here we're actually talking about consumers and, importantly, the decomposers. And essentially, today's lecture will be about this bit here, the phytoplankton, which is the small unicellular algae floating in the ocean, and the zooplankton. And then we actually go from the zooplankton over to the nekton in the next lecture. Don't worry about this thing here at the bottom. It's just if you actually ever need to actually look it up, here it is. Now, biologists have for a long time been fascinated by how the ocean is stratified. And I'm not actually going to force you to actually remember every single distinction, but it is actually useful to look at some of the real unique properties which actually define each layer of the ocean, because it is very, very different from the top down to the bottom. So the zone we usually interact with is, can I actually get rid of this bit here, is basically, called the epipelagic zone. And the epipelagic zone is essentially a very thin sliver. It's almost, like a little bit of a fat layer swimming in cold soup, if you know what I mean, or a very thin sheet of ice. It usually goes down to only about 100, 150, 200 meters, 250 meters in the middle of the Pacific. And essentially, it is defined by the amount of light which actually penetrates down from the atmosphere. Now, light is not very good to penetrate in the ocean. Most of the ocean is perpetually dark. So we're only talking about 200 meters. And basically, phytoplankton is the main powerhouse and the main organisms in that kind of zones. And we talked about the neritic waters and the oceanic ones. So most, really, of the neritic zone is epipelagic because it's usually over the continental shelf, shallower than 150 meters. Now, the next one is, I show you a little bit of that video because that's the really cool zone where the strangest organisms on Earth almost hang out. And that's the mesopelagic. Meso means there is not really enough light for the organisms of, plants and algae to grow, but there's a little bit penetrating. And all those animals are really iridescent. Here we see a comb jellyfish with very, very long tentacles. I don't quite know why we have to have that kind of really weird thing. Now, this is actually not a true jellyfish. It actually sits one step below in the classification, but never mind. And they can be actually up to this size, some of the really larger ones, 2 meters. And those are actually band of little hairs, and they actually beat in sequence in such a way that the different frequencies create different colors. Now, this is a tunicate, and the tunicate is essentially a barrel out of jelly, and inside that barrel is a mesh which actually filters out the phytoplankton, and they all stick together in very, very long chains. Oddly enough, the larvae of tunicates, and this is just an aside, are the first animal in the taxonomic or phylogenetic classification which have the precursor of a spine. So oddly enough, those animals, believe it or not, are the closest relatives of vertebrates of us, because the larval stage is in evolutionary history quite often a more primitive one than the adult organism. So you can watch this by yourself. So I probably, hopefully, told you everything which is on this slide. So we're talking about roughly about 1,000 meters down, and it's also called the twilight zone. The twilight zone because there's a few photons which go down, and bioluminescence is really the cool feature, as I have just shown you in that BBC video. And of course, the further we go down, now we go into the abyss. Now the abyss, had basically, there's a bit of a, classification difference where you can say, oh, is it the bathypelagic or is it the abyssopelagic, or do we talk about the heteropelagic? By and large, many of those animals here migrate to the water column, so it doesn't really matter. But basically, you can say that everything over 1,000 meters is perpetually dark. And I show you hopefully in about 4 weeks' time. We just had a large video conference this morning. Everything is on Zoom now, so you spend your time sitting at home and staring at the ceiling or into a Zoom screen, and wondering, what are you going to have for dinner? But never mind. Essentially, that's perpetually dark, and you need very, very, strong, submarine lights to actually see anything. But what actually is the fascinating bit is, if you look carefully, the ocean only has a very, very shallow surface layer, but from about 1,000 meters downwards to about 11, 12,000 meters, it's the single largest habitat on Earth by a long, 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 long shot. If you ever have to go on a very boring drive and your kids say, "Oh, are we there yet?" which is of course something everybody does in Australia when you go out west, it's nothing compared to the vastness of the ocean, right? So this is really the largest habitat on Earth. Right. You have been introduced to already those weird terms called plankton and nekton. Pardon me. What do marine biologists mean by that? Oddly enough, both of them come from the Old Greek language, and it basically means those are the drifters of the oceans, and those are the swimmer ones. Look at this big whale shark and little diver here. Maybe it turns around and eats him. Anyways, and essentially, of course, the Scopy boat is very much smaller. There are, we're talking about a few millimeters to a few meters in a few very rare planktonic organisms. Whereas those ones, the nekton, are the active swimmers, and they're generally larger. In the plankton, we find plants and animals, whereas the heterotrophs, or animals only, are the sole constituents which make up the nekton. So basically, nekton is sharks, turtles, whales, fish, squid, seals, and we're going to deal with that in the next lecture. Or let's see how far we get today. Right. Now, here is another classification. I promise you it's almost the last one. And essentially, when you talk about plankton, you can say, well, is it an autotroph or is it a heterotroph? Are we talking about phytoplankton or are we talking about zooplankton? In the zooplankton, sometimes, and it's the only time I really mention it here, we can actually differentiate between what we call, temporary stages, like the larvae, and we're going to talk a lot about larvae in a short little while. And we have seen in the last lecture the barnacle larvae and so forth and so forth. Or is it an organism which never actually has another habitat association and spends its entire life in the plankton? We are talking about a holoplanktonic organism then. Right, here we go. One more video, and that's another cool one, The Secret Life of the Plankton. And it actually goes all the way up to a fish.
How did I get here? Well, it's a stranger story than you might think. I came from a world of drifters, a place few humans have ever seen. The world of plankton. I came from a batch of 1 million eggs, and only a few of us survived. When I became a larva, I moved among other drifters. My fellow plankton came in all sizes, from tiny algae and bacteria to animals longer than a blue whale. I shared my nursery with other embryos and juveniles, from clams and crabs to sea urchins and anemones.
So that's an anemone larvae, and the other one is a sea urchin one. Those are the tunicats which we talked about.
Zooplankton.
That's a Bidovincella, Velella. We meet that in a second.
Are copepods krilled. You could search the world over, but you'd never find a place more diverse than my childhood home. A teaspoon of seawater can contain more than 1 million living creatures. It can be a pretty tough existence, though. Trillions are born here, but only a few make it to adulthood. You may be no larger than a pinhead, but this crab larva is an arrowworm's worst nightmare.
In turn, I should say the arrowworms are, of course, the ultimate eating machine when it comes to smaller
Epic
copepods.
battles between carnivores like these are just one way to get food.
And oddly enough, a big jellyfish is a planktonic organism because they got weak powers of swimming. So they might be, 2 meters across, but they're still a drifter.
Transforms sunlight and carbon dioxide into edible gold.
It's a diatom. It's a golden diatom, and when they get into mass concentrations, as we sometimes see on a beach, what we actually get then is a brown discoloration of the ocean beach. And it occurs in batches, and quite often people say, whoa, we've got, some strange, oil spill, and my phone rings, and I say, no, it's diatoms. So diatoms really, in big concentrations, can do that.
Migration of life on Earth. During the day, I'd return to the darkness where I'd join my bizarre companions. Cannibals, like this sea butterfly mollusk that eats its next of kin.
Ooh, that's kind of tasty, huh?
And comb jellies that beat cilia like rainbowed eyelashes. Some of these snare their prey with sticky tentacles, while others just take a bite out of their cousins. And siphonophores that catch prey with toxic fishing lures. But my favorite would have to be the crustacean phronema. Its monstrous looks inspired the movie Aliens. It can catch tiny bits in its bristles, but prefers larger prey like salps. With 2 sets of eyes, this female prowls the deeper water. Prey in hand, she performs one of the strangest behaviors in the entire animal kingdom. With body parts from her victims, she delicately assembles a barrel-like home, feeding her young until they can drift off and survive on their own. Best of all, they make the perfect snack for a small fish like me. Here among the plankton, the food web is so tangled and complex, even scientists don't know who eats whom. But I do. At least now you know a bit of my story. There's so much more to me than just a tasty meal.
Well, here you have it. Next time, or well, it's Easter Friday tomorrow. So for those of you who are Catholics, and it's quite hard to be a Catholic nowadays, after the George Bell verdict. But let's not go there. Of course, you will have to actually eat, fish, or you should actually fast all day. So essentially, if you have a snapper, everything basically starts with the plankton. As a matter of fact, when people talk about, global production of plant material, of energy, of carbon, and converting the conversion of atmospheric CO2 into basically organic material, it's all done, by oceanic phytoplankton, or the most part, not by the Amazon rainforest. It's really the oceans who do it. So the phytoplankton is kind of cool because they're single-celled organisms, which basically means they're very small, so they have a very large surface. So nutrients can go in very quickly, and essentially they can double. They're almost like bacteria, very, very rapidly. So if you want a carbon factory, you want actually small organisms, not large ones, which take 100 or 200 years to, basically mature, and then they fall over and rot. And while they rot, everything basically goes up into the atmosphere again because of respiration. There's actually not that many phytoplankton species worldwide, about 4,000. And we have seen how important the dead bodies are for actually forming sediments in the oceans already. You can actually see them with the naked eye, as I have just explained, when they actually form blooms. And it's quite funny, like one of the most famous blooms recorded in history is actually in the Old Testament, when Moses needed to basically lead the Israelites out of Egypt because they were mistreated as basically foreign workers. Does it sound familiar? By the pharaohs. He basically went to what is nowadays the just at the bottom of the Suez Canal. And the sea was red. Now, the Red Sea is not red because there is desert dust flying in. It is actually called the Red Sea because it has frequent algal blooms, natural ones, which basically discolor the water into sort of an orange, reddish hue. Of course, if you get those kind of bacterial or algal blooms in shallow water, you can have actually very, very big problems in terms of paralytic shellfish poisoning. And I can assure you, if any of you ever wants to experience hell on earth, get a proper oyster kind of seafood poisoning. It's fascinating stuff. Anyways, and also blue-green algae, which belong to the bacteria, are just that. They're microbes, but they're not algae. Hang on, there's a bloody bird. Bugger off. And it's quite funny. Like, you can actually go to a health shop and you buy, blue-green, algae kind of concentrate, but they're basically bacteria, but nobody would actually eat them. So essentially, the phytoplankton comes in a great diversity of forms. This is just like one of those textbook examples. If you look at them in real... Seriously? Mate? Come off. So those are the choice from lecturing from home. You got magpie larks, and they come in the afternoon and need to be fed, and then they have a bit of a protest if nothing comes forward. Anyways, now diatoms are often the most abundant ones, and they usually have two halves. They fit together like a head box. You can actually go. They're beautifully basically structured. Their skeleton is made out of silica, like out of glass. And when you have literally millions of years of deposition of those skeletons, you actually get basically marine sands formed out of silica. And we actually harvest that silica now, and we make what? Porcelain out of it. So, so if you actually have real china at home, it is actually made out of old dead diatoms, or maybe of dinoflagellates. Dinoflagellates are a bit problematic because they have, this, look at this one. I mean, here's a dinoflagellate. It has two tiny little hairs which beat like this, and that beating of the hair propels it forward. But it was really quite funny. Somebody put it under a microscope, and the dramatic music is really quite fascinating. Of course, it's a plant, so it doesn't actually do terribly much. So, you just have to, live with that. Here we go. Now, radiolarians, acantarians, foraminiferous, different forms. Don't worry about those too much. Now, the next.
Humpbacks engulf hundreds of thousands of them in a single gargantuan mouthful. When the going is good, the whales feed continually.
Right. So what we can see basically is those organisms here, and we have one of those krill, beautiful photograph, are really supporting the large animals of this world and all the fisheries. So every fish you eat is going to come from zooplankton. Now, zooplankton is a little bit devilish for the same reason. If you actually think it is hard to identify insects because there is so many of them, or flowering plants, you have seen nothing yet, basically, before you start working on zooplankton. And I can vouch for that. So if you stare down a microscope and you put 1 or 2 drops of water on it, this is sometimes what can actually hit you. And then you go to the next bit, and it looks like this. And then you go like that. And there's about 20 different species here. And here we got one swimming mollusk trying to eat a lot of diatoms. Here we got a crab, we got a little jellyfish, a copepod, another copepod, arrowworm, larvae of some other mollusk, and here we got a quadrumetuse. Here we got the larvae of a crab, and so forth. And essentially, there's about 20 species, and it's such a diversity that it is fascinating on one side, but it also can be demoralizing, and I can vouch for that. The reason I'm showing it is sometimes diversity, we all love diversity as biologists, but when I started my PhD, it wasn't like today where somebody, holds your hand more or less for the next 3 or 4 years as a student. I was literally given the keys to a Land Rover, the keys to a boat, 2 nets, a few buckets, and then I didn't see my supervisor for the next year. He said, off you go, and you do something interesting on plankton. I basically ran and pulled a map, and I thought, oh no, here's an estuary and so forth. But you almost started to cry when you actually start having your first samples, because the diversity is so overwhelming. But then you actually learn that diversity is also quite fascinating, because when your colleagues say, oh, I caught about 20 different types of fish in a day, you say, well, I got about 250 planktons in 1 liter of water. So it is very, very diverse, almost hyper-diverse, and it's also very beautiful. And after a while, you get to know the animals, and it can be quite, almost relaxing counting them. Now, in the zooplankton, there's a big distinction often between what we call the crustacean plankton, like the hard-shelled plankton, and the softest squiggly, plankton. And this fellow here, it's a copepod, and it swims with its antennae like this. Basically, it beats the antennae, almost like a butterfly swimmer. And it is said that this particular fellow here, Gelatinous finmarchicus, is the world's most numerous or abundant animal, by a long shot probably. They usually, in terms of numbers, crustaceans dominate the plankton. They usually have a hard exoskeleton, i.e. they're not jelly. They, think about peeling a prawn. The typical size range goes from 1 to 5 millimeters up to 5 centimeters. Oh, talking about peeling a prawn, I probably need some volunteers to peel lots of prawns, and you can actually eat the rest once we're actually allowed to do field work again, because we're going to do an experiment on whether foxes and birds of prey in dunes on campgrounds eat different bits and pieces of anthropogenic food being left behind, but that's for another point. Talking about which, you will have actually gotten the first set of papers on habitat selection, and I'm working my way through a selection of crows and anthropogenic food, and also of raptors. So that will be with you in a very, very short while. Usually the size range is about 1 to 5 millimeters for copepods, which is quite okay. And Euphausiid squirrel can be 5 centimeters. The problem is they're usually denser than water. Oh, I got hiccups, which basically means they have to continually swim because they sink. Now here's just sort of a selection of smaller crustacean plankton. We got a krill here, which basically forms the food of many of the sei whales in Antarctica, a copepod. This is quite fascinating. I talked quite a bit, and I showed you a video that once you actually lift a bit of kelp from a beach in kind of colder waters, there's often tons of amphipods underneath it. But there's some amphipods which actually have decided, well, bugger this, I actually want to be free. I want to swim in the plankton. And what they did, they basically developed these enormously large eyes. And almost like some weird creature from a science fiction movie, Captain Helmet, with very, very large eyes going about it. Now here's an odd fellow. They're called mysid or opossum shrimp. Opossum shrimp because they breed their young. And I'll show you a little bit something quite funny about opossum shrimp. Here is, I love this picture here, just a little bit of an anecdote really. This is a copepod. So think about a prawn. This is the front part of the body, which you normally, discard. Same kind of things in it. All this yellow, orange, structures is the gut, and the liver actually spills out into little sacs into the body segments. We got 2 short antennae. We got a singular eye. This particular species is called a cyclops, and a cyclops was called in Greek mythology the figure, the monster which lived in a cave, which just had like one eye and blah blah, and devoured everybody coming in. And then the gut goes out towards the back, and out here comes that somebody said in the first lecture it's a turd, and that's what it basically was. But look at this one. Those are the eggs, and the eggs actually got fertilized, basically by another male. And here they were just ready to, burst, and those little nauplius larvae actually swim out. The nauplius larvae will meet again. This is, of course, the one where we actually talked about the barnacles sitting on the shore, blah, 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 blah. Quite cool. Now, copepods, they come in an astonishing array of colors, of forms, but it's basically a breeding machine. It's a feeding machine at the front. And look at the ovaries. Now, think of any other animal you can think of, or think of an animal which would actually have the same proportion of ovaries, of reproductive organs in relation to the body size. I can't think of anyone. It's extraordinary because they are such efficient feeders on the phytoplankton. And that's why it's so abundant and important for food chains. It's basically just a reproductive breeding factory. Some of them actually look a little bit like they have a pajama on, but never mind. And look at this one. Somebody, it plays for about 5 minutes, so I'm going to speed it up a bit. They filmed a copper boat. They fed him some algae. And then out comes the copper boat. And it goes on and go like, oh my God, look at him. How exciting. Yep, and that's about it. And then it floats off. And this is basically what we have seen. It all comes together in the first lecture, which basically transports carbon and bacteria and phytoplankton from the top of the ocean, from the topmost layer down to the deeper ones. Now, this is a plate, a color plate, drawn in ink and then colored in by a very famous, German naturalist called Ernst Haeckel. It's about 150 years old. And some of those copper boats, they basically build out their appendages into the most extraordinary features because that actually prevents them from sinking, so they can actually float. And you might actually look at this and think like, whoa, this guy was basically psychedelic. He must have been on some really good shit, when he drew that. But it's actually true. They are colored like that. So fascinating kind of stuff. Now I put this one in here, the copper boat or arthropoda. And when I was sort of a PhD student, everybody needs to make money. So you work, in hospitality. I got kind of quite lucky. I got to drive, the university boat and do all sort of cool shit. But, I need more money. And then my supervisor said, oh, can you identify some plankton samples, which somebody collected? I said, sure. And those were basically really, really, really tiny copper boats. And I went to the library and I got out a book from 1760. This is not a joke. Somebody went to South Africa, took basically plankton samples, preserved them, and worked them up back in Vienna. And those are the drawings. And I worked from that, and it worked perfectly fine. And otherwise, I would have had no idea, and nobody knew, and I just basically discovered this old publication. So here you go. Sometimes you never know what it actually is used for. So here are those planktonic copepods. Look at the size of those eyes. Extraordinary. And basically, they float at the surface of the ocean. And essentially, they have to escape predators very quickly. They have a brood chamber as well, but the vision is extraordinary. Now we have seen Phronima already. Krill, of course, occurs in big, big swarms and actually underpins Antarctic food webs. Now mysids, why do I show you mysids? Because I know a thing about mysids because that was actually the topic of my master's and then actually of my PhD. So I originally, it's quite funny, that now I work on foxes, eagles, and deep sea corals mostly, but I originally come from a zooplankton background. And mysids have something very, very odd. You see this big, big blades. They're basically, if they have, if you think about it, that's their carapace, and they grow big blades on the side of their body, and they hold them like this, and the young are being brooded in between those kind of brood blades. And they basically produce not larvae, but little mysid shrimp. So you can actually see them in here. And that's why somebody called them opossum shrimp. But what makes them really strange is this. So those were my first 2 papers I ever published. Because what a mysid has is, in the tail here, it develops those kind of weird structures. They're called statolites. And people puzzled for a long time, and they're quite prominent. You can actually see them. And they have little holes in it, and little hairs go into those stones. Look at those hairs. And this is actually my most famous electron microscope, basic image ever. It took me probably weeks to produce this. Those hairs, as you can see, those are 10 micrometers. So each of those hairs is probably 2 micrometer, a thousandth of a millimeter. And you had to dissect those stones out with very fine glass needles, which you made by pulling glass over a flame and then hope it actually breaks in the right sequence. They're arranged in a certain way, and actually the arrangement is specific to species. And you might think, what do they do? Well, they sit at the back, and when the mysid actually swims that way, the stone, that particular structure, pulls on those hairs, and a signal goes to the brain and says, "Oops, mate, you're going too far to the left." So the mysid starts actually beating the legs like this and swims like this again. If it goes too far this way, the hairs pull that way, and the mysid starts to right itself. So they're basically the organs which are the sense of stability or equilibrium. Now we have exactly the same, but ours are in the ears. In each of our ears, we basically have hemicircular canals. When I twist my head like this, water in one of the canals would have actually rushed onto sensory hairs, and now they're like, whoop, I better go up again, or at least I know my head is bent to the left. And we do not know why those organisms, those mysids, have those stones in the tail, but it works perfectly fine. Now, other crustaceans, they actually have their statolites in behind the antennae here, and they shed them during every mode. And then, let's say a little lobster has to go around, or a crayfish, and then they actually pick up little sand grains and stuff them behind the antennae. And then the hairs grow into a little jelly mess, and that actually provides them with their sense of equilibrium. Now, I shouldn't really tell you that. Don't try this at home, but, well, whatever. It's an invertebrate. So you can actually make yourself a remote-controlled crayfish. And all you have to do is basically, you wait until it molts, or you give it particular hormones, then it actually sheds it, but never mind. And then it actually grows a new exoskeleton as it actually grows bigger. And then there comes the time where it says, ooh, I need some sand grains because they need to go behind my antennae so I know whether I'm left or right. Right, and you don't actually supply in the aquarium sand, you actually supply little iron filings, just normal iron filings, which you file off, from a piece of metal. And then basically, same as sand grains, the crayfish stuffs them in, and then you wait for a day until it all grows nicely back together. And then you can approach your aquarium with a magnet. And you can literally make your crayfish run and swim in any odd direction as you like. It's the perfectly remote controlled, basic animal, and it needs absolutely no electronic whatsoever. Oh shit, that actually goes up on YouTube, so I haven't really told you that. I can probably edit it out. Never mind, off you go. So what is of course very interesting, that each of those mysids has a very, very unique stone. They make them themselves. They don't actually precipitate them or borrow them from the external environments. We don't actually quite know how they're so specific. And and I basically did a study where I carefully selected them all out and then put them on an electron microscope. I can actually put the paper up there. It's quite boring really, and a lot of very, very detailed work. It's like when you're a watchmaker, and you have to sort of work at night, because every time the tram would actually go past the building, or even a car, the microscope would shake. So I basically had a nocturnal existence for a few months. But oddly enough, zoology can be quite wonderful. A few years later, we were actually contacted by a petroleum company who actually explored gas and oil reserves in the Black Sea. Because the Black Sea is, of course, full of those mysids. They are literally dominant biomass there. So are they in Antarctica. The grills feed actually on them. And when they actually die en masse in their millions, the stones, those ear stones here, don't rot. And you can actually now geologically date different layers based on different accumulations of different species ear stones. So maybe I should have joined the petroleum company, but here's my contribution to basically, climate, change in a really weird sense. So we should have actually charged them for it, but it's quite fascinating. So here is, of course, the Nauplius larvae again, which we have already seen in the context of talking about barnacles. Right, I showed you that image, and those are all the different stages of a crab, like a shore crab or a ghost crab. And you can actually see it goes through 11, I should say, different stages. And some of them look nothing like a crab. And it is actually fascinating why an organism would spend that extraordinary amount of larval development complexity. We don't really know, but it actually is very, very fascinating. Here I show you some of those crab larvae. This is, of course, the very last stage of the ghost crab, when it actually comes in from the sea and actually settles on the beach again and buries down. And those are some earlier stages of megalopus. Now, we have already seen the lobster larvae. I showed the lobster larvae to you in the last lecture. So this is the front here. We're talking about the gills. This is the back body, and it has 5 legs. This is basically the tail. We're talking about 2 stalked eyes and antennae. Here's another one from a Moreton bay bug. This is the front. Here are the eyes. What is actually quite, look at this video here. It's a Japanese one. Look, does it actually work? Where we actually see one of those big lobster larvae riding a jellyfish. Oh, come on. Be a good boy. No. Hang on. This is the problem, doing, those videos from home. Come on. Right. Maybe you're running out of bandwidth on the Zoom. Is it going to work? No? Is it going to work? Right, no luck. You try it at home. Sorry for that. Now, the next one. Come on, Jesus. We're gonna get there. Right. We go here. Then we go down. Yeah. Now those are my all-time planktonic favorites, or favorite planktonic organisms, and I just let them tell the story. But just look at the beauty of those organisms. Literally, the aesthetic appeal is astonishing.
Allowing them to take advantage of the Earth's largest habitat, the open waters between the sea's surface and bottom. Pteropoda, the Latin name for these snails, means wingfoot. They are planktonic animals, so they are carried along by ocean currents. The pteropods are a widespread and diverse group and play an important role in the oceanic food web. There are 2 distinct types of pteropods: those with thin, lightweight shells and those that have completely lost their shells. The shelled pteropods produce a net of sticky mucus to trap marine snow, material made of dead plant and animal remains, fecal matter, sand, and dust that falls from shallower water. These particles are constantly sinking through the water column, and there is still plenty of nutritious material contained in them. The ability to passively collect your next meal in a drifting net is a great energy-saving adaptation to living in the open ocean. On the other hand, the pteropods without shells are all carnivorous, and as far as we know, feed solely upon shelled pteropods. They have sets of curved hooks and suckers, or sticky tentacles, that are tucked inside the head when they are not actively feeding. When they contact a shelled pteropod, these feeding structures come out to grasp and pull the prey out of its shell. We do not yet know how these pteropods locate their prey, though given that they don't have eyes, they likely use something akin to a sense of smell, or they swim around until they happen to bump into their favorite snack. Pteropod shells are made of calcium carbonate, the same material many marine organisms secrete to form shells, such as clams, or hard supportive skeletons, like corals. There are different ways to produce a calcium carbonate shell, and unfortunately for pteropods, their shells are very sensitive to changes in ocean chemistry. As the acidity of the ocean increases, the fragile pteropod shells are vulnerable to dissolving, challenging the health and growth of these species. This could spell bad news not only for pteropods, but also for the variety of animals, including fishes, sharks, squids, and marine mammals that eat them. The wide diversity of pteropods is a fascinating example of how animals can evolve to take advantage of a habitat where you would never expect to find them. This is Stephanie Bush at the Monterey Bay.
Right. And the next one is very important to watch because essentially this is the world expert on jellyfish, and I could actually make up to tell you something amazing, but Steve Haddock has basically spent his life doing exactly that. And this is a fantastic summary in 5 minutes about jellyfish. And I'll show you some local examples after we have done that.
When conditions are right, these babies can grow up and form vast blooms of adult jellies. But for every moon jelly pulsing in coastal waters, there are deep-sea relatives which defy generalization. They can be a meter across, 20 meters long, and lack tentacles. The branch of the tree of life called Cytozoa also includes another lineage, the coronate medusae. Typically found in the deep sea, they can be brightly bioluminescent, and many lack a polyp stage in their life cycle. All of these cyphozoans are cnidarians, the sea is silent, relatives of sea anemones and corals. This group is defined in part based on the presence of stinging cells. The most infamous stingers are the sometimes lethal box jellies found in tropical waters, but most cnidarians pose no danger to humans. Most species of swimming jellies are actually in another group called the hydromedusae. These are often small and transparent. They may have very few tentacles, or very many. They might or might not have a small polyp stage called a hydroid. One group of hydromedusae has tentacles which point ahead of them instead of trailing behind. These eat other gelatinous organisms, rather than the crustaceans favored by many other cnidarians. But even with all this diversity, we haven't yet encountered the most unusual hydrozoans, which you consider a siphonophore jellyfish. From the Portuguese man-war floating on the surface, to the dozens of species found in the deep sea, these are among the strangest of marine animals. They have divided up the tasks of living among different subunits. Some parts pulse to move the animal through the water, but they can't feed. Others, connected by a thin tube that runs the length of the body, can feed or reproduce, but not swim. Although siphonophores can grow to be 30 meters long, the most common are small, rocket-shaped species found throughout the ocean. Those are the cnidarians, including some of the most familiar and unfamiliar animals that might be called jellyfish. They all have in common the presence of stinging cells called cnidae. Not all jellies sting, though. Another deep branch in the tree of life leads to the ctenophores, also known as comb jellies. Their defining trait is 8 rows of ciliary plates, which flutter like eyelashes to move them through the water. Some species use sticky tentacles to capture prey, much like a spider web. Others lack tentacles, but use tooth-like cilia in their mouths to bite and swallow gelatinous prey. Many deep-sea species of comb jellies are so fragile that they are new to science or have only been recently described since the advent of submersibles. Nearly all are bioluminescent, and many have red or black pigmentation, which is thought to mask the luminescence of their ingested prey. The next group that is often lumped with jellyfish are urochordates, including salps, doliolids, and larvaceans. These are the most vertebrate-like of invertebrates, with many traits shared with chordates. Salps are typically colonial, forming units that can be tens of meters long. Each unit within the colony pumps water through their body, filtering out phytoplankton and even smaller particles. Therefore, they are the only jellies that subsist directly on algae and microbes. Because they grow and reproduce rapidly, salps and doliolids can form dense blooms in many regions. Larvaceans filter particles using a mucus house that they secrete and inflate, sometimes many times a day. The mesh is so fine that they're able to retain and ingest even bacteria-sized particles. So the factors that lead to changes in salp populations and the effects that salps have on an ocean ecosystem are very different from the causes and effects of changes in cnidarian and ctenophore populations. We know of slugs and snails that crawl on the ground, but a variety of mollusks live in the water column, flapping their feet to swim. Many have even retained their shells, although typically in a reduced form. Many kinds of worms also live a gelatinous life. This includes the polychaetes, which are distant relatives of earthworms. One of the most abundant deep-sea animals, below 2,500 meters, is actually a polychaete with bands of long setae, the bristles that characterize polychaetes. Other segmented worms, transparent and beautifully bioluminescent, lack these bristles entirely and swim around, preying on gelatinous organisms. Some worms, with 2 beady red eyes, are parasites and predators on jellies. So is there really such thing as a jellyfish? Are they only circular cnidarians or also elongated ones? Do they sting people or eat microscopic plants? Are they rare and elusive or poised to take over the world? All across the tree of life, organisms have evolved jelly-like adaptations, and each has its own way of thriving and surviving. Understanding their diversity is an important first step to understanding life in the largest habitat on Earth. This is Steve Haddock from the Monterey Bay Aquarium Research Institute. If you see any jellyfish-like creatures, be sure to report them at jellywatch.org.
Right, so that was a really fascinating insight into the gelatinous zooplankton. And the gelatinous zooplankton, come on here, you're a good boy, right here we are, is a little bit of a problem because most people don't really like jellyfish because they either sting, and of course in Australia we have a huge problem, I wouldn't say problem, yeah, well, we do, with stinging jellyfish, and or they're kind of large and unsightly, or they're just really, not what the general public, finds terribly appealing. However, if you actually take all of those anthropomorphic feelings out of the assessment of jellyfish, the one thing I have to say as a biologist, and even after 40 years actually working on that kind of stuff, what really, really fascinates me is that how such a very, very primitively organized, basically, life form can be so incredibly successful. And perhaps it is the simplicity of how they're actually made, being essentially a jelliness, where the gut is in the middle, and then you have a few tentacles to capture stuff, which may be key to the success. But what they can also do is, we talk about leafcutter ants or about termites or other social insects such as bees. But jellyfish have actually, solved the problem of a colony, in the most extraordinary way, and that's the Portuguese man of war in Australia called the blue bottles, because blue bottles are essentially a colony of different, basically jellyfish, where each individual takes on a different role. Yeah, you have seen some images. Those are classic deep sea jellyfish. This is sort of box jellyfish from the north of Queensland. What most people don't actually know, there's about 14 or 15 different types of jellyfish, which cause what is called the Okenji syndrome. And how do I actually express this now without getting into really, really deep trouble? It is basically, as we heat up the ocean through human actions, more and more of those tropical species are likely going to come down. We actually tried to get some money from the government to actually study that, but it didn't actually go all that well. Well, it went to a certain extent, but we haven't really given up, so watch this space. Now, what you can do, you might say, well, I never actually find any jellyfish because I don't have a boat, I don't have a plankton net, and the uni doesn't actually allow us to go on boats anymore. By the way, a few students actually have emailed me and say, look, can I get a discount because we didn't actually go to Fraser Island? I can't actually make those decisions. I'm just a simple biologist at the bottom of the food chain, so to speak. So if you actually want to actually do that, query that, you have to actually direct that to the Deputy Vice Chancellor academic. His name is Tim Vess, so that's the place to go. Anyways, so you might actually say, oh, what is that? But you can actually do a little bit of jellyfish biology by actually walking on the beach, particularly during prolonged onshore winds. Often you find accumulations of those. Nice photograph. I wonder who took it. Anyways, and they have this sail here, and it's called Velella, called by the wind sailor. And those sails are orientated in a certain direction relative to the main disc of the body. They're only about that size really, and they're perfectly harmless. And different populations have different orientations of the sail depending on which ocean they live in. And of course, we have got the infamous Portuguese man of war, or, basically blue bottle in our bits and pieces. We quite often find those ones perfectly harmless. They're called blue buttons, bobita, and this is the jellyfish body, and those are all its little tentacles hanging down, but essentially they capture the tiniest of copepods, even bacteria, so perfectly harmless. Then we got bubble snails, and this is actually a snail which kept its shell, but what it actually does, it actually floats on the sea surface and eats jellyfish. And it actually floats by producing a raft out of bubbles. Quite ingenious, out of slime. I love those kind of things. And then you actually have those things called a sea glaucus, or a pelagic sea slug, which actually eats all of them. So you can almost, on a sandy beach, construct a little bit of a pelagic food web, where you have the vitamin sailor, which actually gets eaten by the bubble snail. Those gets eaten by the bubble snail, and this guy here basically eats everybody. I should probably have put him right at the top. Here's the Portuguese man of war, or maybe not.
This Pelagia noctiluca is a successful hunter, and several small crustaceans have fallen prey to the jelly's appendages. The nerve poison has an immediate effect. Directly under the surface, myriads of mini medusas are drifting by, either towards death or adulthood. Many of them will end in the bellies of loggerhead turtles. Is it a jelly chain? No, it's a colony of Thaliacea. They're not jellyfish, but astonishingly are more closely related to vertebrates, even us.
Lucky.
Pelagia noctiluca is also looking for plankton, just like the Thaliacea. But this lion's mane is relatively harmless in comparison with this Portuguese man of war.
I mean, what we don't actually realize is
but they can sail.
how lucky we are in Australia. I went to the Azore Islands to work there for a while, which is in the middle of the Atlantic, and the blue bottles there are about that size. You have never seen anything like this. It's 20-meter long tentacles. So if you actually think like blue bottles this size are bad, yes, they're not pleasant, but they are absolutely nothing in comparison.
Up to 10 meters. The Portuguese man of war will fire off whole batteries of stinging cells at its victims. It's clearly visible that the medusa stockpiles food, which is essential because there are often days where the hunt yields nothing at all.
Right, we do that on next. So this is the drifting bubble snail, very, very cool, and you can actually see that in our waters. Here. The problem is we're all sharing the same bandwidth, YouTube and the other ones. Right, and in the twilight zone, what I actually said right in the beginning when we talked about the different, stratification of the ocean, this is actually an interesting habitat because there is nothing like this on Earth for two reasons. The first one is you have permanent darkness, and the second one is none of those animals living there will ever have contact with anything hard. They're always surrounded by basically water. They never touch they might touch a seamount, which is an undersea mountain occasionally, but largely they are surrounded by dark water. You might say, that's not very unusual, but it actually, in the greater scheme of things, it is a very, very unusual feature. Now, what is actually odd about the plankton is that you can actually go to any particular spot and you go there during the day or during the winter, and you might find nothing. And you say, oh, yeah, somebody actually told me there's all this plankton around. But plankton, despite by being the drifters of the ocean, they have two peculiar, modes of distribution changes. The first one is they have cycles where the mature females go and move to the top of the ocean, the surface of the ocean. They breed there, and the larval stages go to the bottom of the ocean where it's dark, they mature, and then they develop into mature females again. And the other one is the reverse of that cycle. However, what's going on there? It is actually the second behavior, which is much more intriguing and widespread. And I'm playing you a video now, not only because I think it's excellent footage, and every biologist should actually see that, but there is a message in it. And I wonder if you can actually see what the message is in relation to plankton. You can actually watch a longer video of that on a BBC channel and in better, resolution. And watch the next one. That's the really cool footage. So the reason I show you a lion video is not because I'm particularly bloodthirsty, but as a biologist, there is a lesson in that. When, until about, 20 years ago, you actually read a book, there's actually a very famous book about hyenas called The Hyenas of the Serengeti by Hans Kruk. And hyenas are described as basically being scavengers, and they eat the remains of whatever the lions leave over from a big kill. At the same time, you basically can go to a game reserve, and mostly people, and I know I used to work there, people, basically want to see a lion. And I always say, let's look at something else, because lions are, by and large, for the most time, the most boring animals on earth. They just basically sleep 23 hours and 50 minutes a day because they don't want to actually spend any energy. However, and this is the lesson here, life on earth in any organism works 24 hours a day. It happens 24 hours a day. And essentially, if you don't prepare to become a nocturnal observer from time to time, you're actually missing very, very essential parts of the story. So when we actually started to look at lions and hyena, we actually changed our pictures, and you have to actually, hyenas get killed by lions, which we didn't really know. But what you also didn't know is that most of the kills in Africa, actually of large game, happen by hyenas as active predators and not as scavengers. And again, that actually happens usually during the night. So essentially, you can only piece together a full story of an ecosystem or of a particular fauna component if you look at it 24 hours a day. And you might say, now, well, what has this got to do with, the plankton? Well, even though, we actually think that, plankton is everywhere, and it is actually very abundant, you can actually get the best plankton gathering equipment, and you can go to a perfectly nice estuary or out to the ocean. And if the sun shines brightly and you go there, you find bugger all. You might get a few grab larvae, maybe a few arrowworms. And this is exactly what happened to me. I was so naive. And I drove with the boat up and down, to different spots. And I thought, hang on, why don't I actually catch anything? I thought maybe I'm not operating the nets correctly. I go too fast, I go too slow. The point basically is most of the plankton during the day sits right at the bottom. And, we got a dog. And they sit at the bottom here. As you can see, this little, student here. And essentially, if you want to survive, you only go up. Who has the dog? Can we see him? No? All right. Somebody must have the microphone on. So if you have a nice dog, let's sort of share him, I'm actually putting in, I've actually put in for an adoption dog, so let's see how that goes. Anyway, so maybe next time, he can actually give the lecture instead of me, or sit here. Yeah, that would be kind of cool. We could actually all have like a dog Zoom, huh? That would be really cool. Never mind. So essentially, the plankton have a very peculiar behavior in that they switch habitats between day and night. Now, how does this work? Essentially, even the old naturalists knew that. You see the sun, going down, and it becomes crepuscular night, then it's actually midnight, and then it actually rises again, and in the morning it dawns. And so essentially, the plankton goes to the surface, and you might actually catch it, then it gets dispersed again, and then it goes down again. You might say, come on, somebody actually just made that up, because they threw up those little mysids here. And here's another one. Basically, at dusk, you go up, then you stay at the surface all the time, and then you go down. But what we actually do know now, we can actually measure the particular light level which makes plankton migrate. It's called the isolume. So essentially, based on the isolume, you can either go up or you can actually go down, depending what the light level is. Now, it's a very, very regular occurrence, and there's two different types of hypotheses out there to explain it. The one is called the growth rate hypothesis. Now, the growth rate hypothesis basically says that you cannot basically, feed and digest continuously because there's limits to the food supply, so sometimes you actually have to stop feeding. That is actually problematic, because if you don't actually feed, you lose energy. We do the same thing. As I'm actually sitting here, and talking to you, and you might actually sit on the couch and have a sandwich, or a chocolate or a milkshake or whatever, is basically I lose energy because my basic metabolism carries energetic costs. So what you might actually do is, because the deeper layers of the ocean are colder, you could actually derive a benefit for actually staying in colder waters when you're not feeding, migrate to the surface, feed, and then go down again, because that slower growth basically translates into longer lifespan, and longer lifespan means a greater probability of producing young. And of course, you want to go into the surface layer because that's where all the productivity happens. So that's essentially where you can exploit the rich food sources. So what we have here is a migration based on energy expenditure. The other one is much better. It's called the death rate hypothesis, and it's quite simple. It basically says the predators of the plankton, being mostly fish, they hunt by sight. So when you should actually go up into the sunlit layers, it should be during the night when the fish can't actually see you. You can still gobble on the phytoplankton, but as soon as the sun rises, you have to go down because you expose yourself to predators. Seems to make sense. Now, how can we actually prove this? One of the most elegant experiments, which of course nobody knew about, comes from lakes and the Alps and monks. And you might say, oh, now he's actually losing it completely. Well, here's a monastery, and that particular monastery called Grimsminster, used to own most of the land, which we know as our family grabbed from them from, I don't know, some shady deals, whatever happened there. But they also owned many of the alpine lakes. Why? Essentially, here you go, here you have a fish, and during fast times, and particularly during Friday, the monks were only allowed to eat fish and not meat. So they had a very, very peculiar and very strong interest of actually having the best, fish, coming only exclusively to them. And so they basically, appropriated lots of lakes. And some of those lakes, so this is the lake which actually now belongs to our family in the Austrian Alps. And it used to belong to a monastery. And they carried little fish larvae up there in the 16th century, and we got very good records of that. And basically those fish actually grow. They don't actually grow very fast. They're basically Arctic char because it's very cold. And essentially what happened is, and this is kind of cool, is they didn't get to all of the lakes because many of those lakes are naturally fish-free because the fish never actually got there after the last ice age. So half the lakes are, of course, without actually any fish, and the other ones have. So you can actually go and take a plankton net through that, and I have done that, with some of my colleagues. And basically, this is a lake with fish. During the day, it has fish for 300 years. You find no plankton; they all sit right at the bottom. You go across, that little sill here. There is another lake, and it has no fish in it at all, and the plankton is plentiful. They don't migrate. So this is actually a very, very cool experiment done by monks in the 16th century, and we actually take advantage of it to test the growth rate hypothesis. So that behavior called the daily vertical migration is something quite extraordinary, and an enormous amount of energy and time is allocated to it. And the vertical ranges are also extraordinary. Some of the copyboats which are a millimeter, they might actually go 10 to 20 meters per night. The mysids, they go up to about 300 meters. And some of the small fish or fish larvae, they can actually go to a kilometer up and down every night. So this particular small fish basically holds the record. It's about 6 centimeters long, not very, very large. And it sits around 16 to 1800 meters. And every night, basically, it goes almost to about the bottom of the epipelagic zone and then down again. So it's about a 3-kilometer round trip. So if we basically scale that up to our body size, it would actually mean we run a marathon every day just before breakfast. So the predation pressure, the evolutionary pressure to actually do that must have been quite extraordinary. Now, the feeding of zooplankton, we have seen a few things already, and I very quickly go through that, simply because the zooplankton are so important in terms of packaging carbon produced by phytoplankton for higher trophic levels. We have things like the arrowworms, which we saw, which are essentially just like this one here. It's two big jaws, two big eyes, then a very, very agile body which can dart forward, and they basically prey on the copyboats. Then we of course have the various forms of jellyfish, and they basically have the stinging cells. You will actually know that from the zoology lectures in year 1. It will be actually quite interesting how we're actually going to do all the sort of breaks. Will I actually sit at home then with a jellyfish here on my lap and show you? Time will tell. And then the herbivorous copyboats. The copyboats have been called basically the grazing bisons, or whatever you want to call them, of the ocean. And the ecological role in a global ocean is to package primary productivity from phytoplankton and essentially put it into bigger parcels, which fish larvae, bigger zooplankton, krill can eat. And so if you actually look at a copyboat from the bottom, essentially it has a few legs here. This is the tail. This is to actually stabilize them when they swim. Those are the antennae which they use to basically get forward in the water. But most of the mouth parts here is just a gigantic feeding structure there to actually capture phytoplankton. And so essentially they can graze anything up to about 1/10 of their body size. There's some size selectivity, but they're often called the sheep of the ocean. I just call them the bisons. It kind of sounds a little bit more dramatic. So they're the major herbivore group. And they can either tart at particles, actively and actually grab them, like you would actually catch a cricket ball, ideally speaking, one which isn't tempered by, sandpaper, but let's not go there. Or they basically can just sieve it out, from the water column, almost like actually taking a strainer through it. Now, those are the coolest ones, which I actually quite like. Those are the appendicularians. Never mind, we actually saw them. And what they can do, here we go, is they can feed down to particles the size of,
Giant larvation.
bacteria and viruses. Because
The animal is actually the blue tadpole-looking thing.
this is the little
This is the
animal.
body.
Oh, here we go. We got
And this
an
is
error.
its
Hang on,
tail.
let me get out of the way here. And
Bathycordius is considered a giant among larvaceans.
and it
Take a
actually
look at this one.
beats the tail,
It's
and
about the
that
size of a
creates a
breakfast
water
muffin.
column, a water
But
current.
it towers beside the tiny, regular variety of larvation. I know they don't look like much, especially in the deep midwater, where you've got bright, colorful jellies, big, cool eyes, and shimmering surfaces. The competition for weirdness here is tough. Take the silky medusa, for example, which can dump all its tentacles as it swims away. Or this vampire squid, which appears to turn itself inside out. Larvaceans can't do that. But what they can do is downright incredible. The giant larvacean's claim to fame is the huge house it builds. I'm talking about that big mucus bubble that surrounds the larvation. That floats the larvation. Larvaceans are
So that drags on a bit, but basically they build a gigantic mucus house which can trap bacteria and viruses, up to those kind of things. And then every now and then they eat their own snot, which is actually loaded with all sort of goodness. So if your kids ever pick their nose, then just tell them that. So why do we actually spend time looking at plankton? And there's just every singular big oceanographic research institute in the world would have a very, very good complement of plankton researchers, simply because if you don't actually understand plankton, you have actually no hope of actually understanding fisheries and regulating it and conserving it. Because ultimately, all food chains, communities, species in the ocean are underpinned by the primary productivity and then by the trophic packaging up a cascade by the plankton. And it doesn't really matter, which diagram you have. You can actually have it very, very simple. Here you have a few diatoms, and they get eaten by krill and then straight up to the rails. Or it can be a little bit more complicated, but the essence is the same. Somebody in this big blue yonder out there needs to capture the carbon and make it into a biologically digestible and usable form. And this is basically the zooplankton. And then somebody needs to package those small little phytoplankton cells up so they basically can be eaten by larger vertebrates. And that's basically what the zooplankton does, like here, 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 here. And then it actually goes up to, what we actually call nektonic organisms: whales, seals, birds, fishes, even penguins, and where are sharks, and here's a petrel, and so forth. So essentially, all of those vertebrates which have much better movement abilities compared to the plankton. So ultimately speaking, what we have talked about today is the foundation of what we're going to talk about next time, which actually deals with those kind of very, very lovely, organisms, which basically are the predators of the ocean. Right. This is the lecture for today, and next time we talk about the nekton, the big consumers